Hello there. In this video, we are going to discuss about the mechanism of cell injury and cell death, and different factors which are responsible for cellular death. So it is going to be a large topic because we are going to cover multiple factors which are responsible for cellular death. So let's start it. So first of all, now we have to consider some facts related to cellular injury. The first one is cellular response to injury stimuli always depend upon the type of injury, its duration, and its severity. If there is a more severe injury stimuli, then there will be direct cellular death. If it is less severe, then there will be reversible injury, and so on. Next one is the cellular response also depends upon the type of cell and its genetic makeup as well as its adaptability. For example, the skeletal muscle cell in the leg can tolerate ischemia about 2 to 3 hours, while cardiac muscle cannot tolerate so much. Skeletal muscle dies in 2 to 3 hours of ischemia, while cardiac muscle dies in 20 to 30 minutes of ischemia. So the response of cell to injury stimuli also depends upon the type of cell and its adaptability, its genetic makeup, and its status, nutritional status. For example, a glycogen replete hepatocyte can sustain hypoxia and can bear hypoxia for longer duration as compared to hepatocyte which is depleted of glycogen and any glucose storage. Next thing is cell injury usually results when there is functional and metabolic and chemical derangement in cellular function. When cellular function and cellular components are altered either functionally or metabolically or chemically, then there is cell injury and cell function is lost. And different type of cell injury produce different type of responses in cell. For example, hypoxia and ischemia lead to necrosis, while when there is damage to DNA or there are misfolded protein, apoptosis is mostly triggered. However, these mechanisms are mostly overlapped. Injury can stimulate necrosis as well as apoptosis or both at a time. So next we move towards different factors which are responsible for causing cellular injury. So we will be studying about hypoxia, ischemia, ischemic reperfusion injury, and about oxidative stress. And then we will move on towards cell injury caused by toxin, cell injury caused by endoplasmic reticulum stress, and then we'll move on towards DNA damage, inflammation, and common events in cell injury, mitochondrial dysfunction, and defects in membrane permeability. So we have to cover a lot of topics in today's video. So let's begin with the first one, which is hypoxia and ischemia. So what was hypoxia? Hypoxia was loss of oxygen, and ischemia was deficiency of blood. Hypoxia, deficiency of oxygen; ischemia, deficiency of blood. So what happens most of the cellular activities are dependent upon ATP. So when there is hypoxia or ischemia, ATP production declines first because oxygen is the final acceptor of electrons in electron transport chain of mitochondria. So when there is no oxygen, electron transport chain fails. When electron transport chain fails, ATP production declines. When ATP production declines, ATP dependent processes of cell are retarded. So our healthy human cell burns about 50 to 75 kg of ATP in one day. So from here you can appreciate the importance of ATP in the cell. When there is no ATP, there is drastic effects on the cellular activities. So how cell cope with this hypoxia? Many cells in the body have capability to cope with hypoxia by increasing the production of vascular endothelial derived growth factor and increasing the uptake of glucose and upregulating the enzyme of glycolysis. And these changes are induced by a family of transcription factors, which is hypoxia-inducible factor 1 family. These transcription factors move through DNA and they increase the transcription of messenger RNA, which leads to production of vascular endothelial derived growth factor and enzyme involved in glycolysis and proteins involved in glucose uptake. And overall, there is better response to ischemia and cells survive the ischemic condition. However, if ischemia continues for indefinitely, cell can no longer survive until leads to cellular death. And further here, the cells which are contain glycogen or glucose storage such as liver and striated muscle or skeletal muscle can tolerate hypoxia and ischemia better as compared to cells which don't have these storage such as neurons. And some cell, even in the presence of oxygen, they carry out anaerobic respiration. And anaerobic respiration also a way to tackle hypoxia. A cell shift from aerobic respiration to anaerobic respiration in which 2 ATP are produced and in the end pyruvic acid is converted to lactic acid. So many cells use this mechanism also to cope with hypoxia. However, cancer cells and rapidly dividing cells, even in the presence of oxygen, carry out anaerobic glycolysis. So why they carry out? Because they use the metabolic product formed in this process as well as the intermediate of TCA cycle for synthesizing their cellular components needed for their rapid division. So this phenomenon is termed as Warburg effect. So what is Warburg effect? Cancer cell and rapidly dividing cell, even in the presence of oxygen, carry out carry out glycolysis. So they obtain energy from glycolysis instead of obtaining energy from electron transport chain and TCA cycle, and they use the intermediate of glycolysis and TCA cycle for synthesizing the lipids, protein, DNA required for their continuous division. So this phenomenon is called Warburg effect. Next, move on. What hypoxia and ischemia do to the cell? When hypoxia ischemia, there is no ATP production. When there is no ATP production, ATP dependent sodium potassium channels are blocked. When they are blocked, sodium accumulates into the cell, and when sodium accumulates into the cell, there is osmotic gain and water enters into the cell. As a result, cell swells up, and this leads to cellular injury. Moreover, there is anaerobic glycolysis. When there is anaerobic glycolysis, there is accumulation of lactic acid. When there is accumulation of lactic acid, intercellular pH decreases. When pH decreases, many functioning of many cellular enzyme is altered. Moreover, when there is depletion of ATP, protein synthetic apparatus also disturbed. When it is disturbed, ribosomes detach from the endoplasmic reticulum, and polysome break into monosome, and protein synthesis in the cell decreases. And protein is a crucial component of cellular functionality. When protein production decreases, cell function decreases, and ultimately can lead to cell death. Moreover, hypoxia also leads to production of reactive oxygen species, because when there is hypoxia, there is mitochondrial dysfunction. When there is mitochondrial dysfunction, the mitochondria cannot cope with the proper oxygen reduction. As a result, there is production of reactive oxygen species, which further induces cellular injury, and the cellular injury also mediated by ischemic reperfusion injury, which is the next topic. Moreover, hypoxia also leads to damage to mitochondrial membrane, lysosomal membrane. Mitochondrial membrane damage leads to mitochondrial dysfunction and release of cytochrome C, which leads to apoptosis. Lysosomal membrane are damaged, and lysosomal membrane are damaged, their enzymes are released, which lead to apoptosis. And the ultimate result of all this phenomena is cellular death, either via apoptosis or necrosis, mainly necrosis. Next topic is ischemic reperfusion injury. So what does it mean? It means here, when there is ischemia and blood supply to ischemic tissue is suddenly restored, what happens? Instead of reversing the injury, there is more injury, there is more cellular damage. How it happens? In ischemic tissue, the capability of ischemic tissue has lost its capability of tackling the reactive oxygen species properly. So when blood supply to ischemic tissue is restored, oxygen level in the tissue increases. When oxygen level in the tissue increases, there is production of reactive oxygen species, because its mitochondria is damaged and it is not capable of reducing all the oxygen properly. As a result, reactive oxygen species are produced, and the mechanism of antioxidant mechanism are also altered and deranged. So the reactive oxygen species are not managed properly. Moreover, the leukocyte also produce reactive oxygen species, which leukocytes are increased in number due to entry of more and more blood due to restoration of blood flow. So this factor lead to production of reactive oxygen species, which induce the cellular damage. Moreover, when there is increased blood flow, there is also increased in amount of leukocyte in the cell, and this increased amount in leukocyte also produce more and more cellular damage, and they will induce inflammation. They also activate complement system, and all these processes lead to more cellular injury. So what is ischemic reperfusion injury? When blood supply to ischemic tissue is restored, oxygen level in the ischemic tissue increases, but ischemic tissue is not capable of reducing all the oxygen due to deranged mitochondria. As a result, there is production of reactive oxygen species, but antioxidant mechanism are also deranged, so these reactive oxygen species not managed properly. As a result, there is cellular injury. Moreover, leukocyte increase there is complement activation, there is inflammation, and all these processes ultimately lead to more and more cellular injury. Our next topic is oxidative stress. So what is this oxidative stress? Oxidative stress basically this stress of reactive oxygen species or reactive free radicals which induce the cellular injury. So these reactive oxygen species or free radicals are produced as a result of chemical injury to the cell, because of drugs, can be due to radiation injury, can be due to hypoxia, can be due to cellular injury, and so on. There are multiple causes of production of these reactive oxygen species inside the cell. But cell have proper mechanism of tackling these free radicals. If these mechanisms are deranged, then there is injury due to these free radicals. Let's move on and study them in detail here. So in general, reactive oxygen species are produced in two processes, which are here. Reactive oxygen are produced normally in small amount in redox reaction. Redox reaction is mainly going on the electron transport chain inside the cell, where oxygen at the end is final electron acceptor. This reaction is not always perfect. As a result, some amount of free radicals are produced here. Which free radical is produced here? There is production of superoxide radical in this process. This superoxide radical is finally converted into hydrogen peroxide via enzyme superoxide dismutase. So at the end of the electron transport chain, some amount of superoxide is produced, which is converted into hydrogen peroxide by enzyme superoxide dismutase. H2O2 or hydrogen peroxide is stable as compared to superoxide, but it is again converted into uh, the OH ion or hydroxyl ion in the presence of some metallic atom. So this reaction is called Fenton's reaction. So this is the production of free radical in the redox reaction taking place in electron transport chain. Reactive oxygen species are also produced by phagocytes such as neutrophils and macrophages, as we also discussed in ischemic reperfusion injury, and how they are produced in their membranes. Superoxide is produced in the same way as in electron transport chain. This phenomena is called respiratory burst, and this superoxide is again converted into H2O2, and H2O2 in turn here is converted into hypochlorite HCl by enzyme myeloperoxidase. The H2O2 produced on electron transport chain is converted into hydroxyl ion, while in leukocytes it is converted into hypochlorite or HOCl by enzyme myeloperoxidase, and which is bactericidal agent. Moreover, there is production of nitric oxide in this phagocyte, and this nitric oxide combines with superoxide and produce peroxynitrate, which is also involved in cell injury, as well as it is involved in antibacterial role. So these are two mechanisms by which reactive oxygen species are produced inside the cell. One is by electron transport chain and another is by leukocytes. So this free radical production is increased abnormally under certain conditions, such as when there is radiation injury by ultraviolet rays, X-rays, when there is injury to certain chemicals, certain drugs, when there is inflammation, or when there is ischemic reperfusion injury, their production is increased. For example, here they say that ionizing radiation split the water and produce hydroxyl radical and hydrogen radical. So these radicals are highly reactive and they induce the cellular damage. And this cell have mechanism to counter these free radicals. What are these mechanisms? For example, superoxide is produced, which is converted into H2O2 by superoxide dismutase. This is also a mechanism. Then then hydrogen peroxide H2O2, it's converted into water by enzyme catalase. Catalase enzyme which combines oxygen with H2O2 and converts into the water. It is also antioxidant mechanism. Then there is a mechanism of glutathione peroxidase. What happens? Reduced glutathione. This is reduced glutathione which combines with H2O2 and it is converted to oxidized glutathione and water. In this way, H2O2 is reduced to water. Then this oxidized glutathione is converted back into reduced glutathione by enzyme glutathione reductase. So what happens? In the presence of glutathione peroxidase, reduced glutathione combines with H2O2 and is converted into oxidized glutathione and water. This oxidized glutathione is converted back to reduced form in the presence of glutathione reductase enzyme. So in this way, cell counters these free radicals. And there are also nutritional antioxidants, for example vitamin A, C, beta carotene, etc., which are also involved in antioxidant role inside the cell. So how cell is countering these free radicals? By superoxide dismutase, by glutathione peroxidase, and by catalase, and by nutritional antioxidants. What these antioxidants do? These antioxidants basically lead to lipid peroxidation, they damage the lipid inside the membrane. When lipids are damaged, membrane permeability is altered. Membrane permeability is altered, then there is mitochondrial dysfunction, there is lysosomal dysfunction, there is cellular plasma membrane dysfunction. As a result, they lead to cellular injury and cellular death. There are protein modification, even protein break, even protein, there is misfolding of protein. As a result, they lead to a functional dysfunction of the cell. As a result, it leads to cellular death. There is DNA damage, there are double strand breaks, there are mutation inside the DNA which can lead to neoplastic transformation, cellular death, or aging. These things are discussed in this paragraph, that they induce lipid peroxidation and membrane damage, cross-linking of protein and protein damage, misfolding of protein, which damage the cell function, ultimately lead to cellular death, DNA damage, which lead to mutations, aging, and ligand transformation, and ultimately can lead to cellular death. Here they also say that these reactive radicals have some role in signaling pathway inside the cell and some physiological functions. However, their production is tightly regulated. If their production increases than normal, then they induce the cellular damage. Next topic is cell injury caused by toxins. So toxins can be directly acting toxin and latent toxin. Directly acting toxins are those toxins which act directly on some of the cellular pathway or some cellular organelle. For example, mercury chloride is an example which inhibits several enzymes inside the cell by binding to their sulfhydryl groups. When they inhibit enzyme, there are different processes in the cell which got inhibited. Similarly, chemotherapeutic drugs which also direct acting play role as direct acting toxin and inhibit various cellular enzyme. For example, methotrexate which inhibits enzyme dihydrofolate reductase and so on. However, we need not to go into their details because toxins are mainly present in forensic portion, and they are also present in pathology in next chapters, and the chemotherapeutic agent basically in pharmacology. So what are latent toxin? Latent toxin are those toxins which does not act directly. Instead, they are activated before they damage the cell by inhibiting some pathway. So they are mostly activated by cytochrome C450 pathway of endoplasmic reticulum in liver and other cells. Here is an example of carbon tetrachloride CCl4, which is used in clinic agent and analgesic acetaminophen. So what happens, CCl4, when it is inhaled and taken inside the body, it is metabolized and converted into toxic free radicals. What these free radicals do? These free radicals damage the hepatocytes. They damage the endoplasmic reticulum membrane. Then the ribosome got separate from the endoplasmic reticulum. There is decreased protein synthesis, apoprotein synthesis decrease, and these apoproteins are part of lipoproteins. Then there is a decreased production of lipoprotein. As a result, fatty acid and lipids accumulate inside the liver, which lead to the fatty liver of CCl4 poisoning. And then there is mitochondrial injury, ATP store decrease, and ultimately lead to death of hepatocytes. So what the CCl4 is doing? The CCl4 damages first endoplasmic reticulum, decreases the production of apolipoprotein. As a result, decrease of lipoprotein decreases. When lipoprotein production decreases, there is fatty liver of CCl4 poisoning. Then there is mitochondrial injury, ATP production decreases, and ultimately all these factors lead to the death of hepatocytes. Next, to move toward endoplasmic reticulum stress. What is endoplasmic reticulum stress? Endoplasmic basically misfolded proteins which are not folded properly accumulate inside the endoplasmic reticulum, and endoplasmic reticulum induces some response due to the accumulation of misfolded protein. This response is of two types. We can appreciate all this. We can understand all this by diagram here. This is the diagram of endoplasmic stress. So this is the lumen of endoplasmic reticulum, and this is the cytosol, and this is misfolded protein which are accumulated here. So what happens when there is mild ER stress or some quantity of misfolded protein? What happens? This misfolded protein bind to the receptor here, which is sensor of misfolded protein, or IRE1 receptor. So what happens? This receptor induces a signaling pathway which leads to the production of increased production of chaperones. So what happens? Misfolded protein bind to receptor. The receptor lead to signaling pathway. This signaling result in increased production of chaperones. What are chaperones? Chaperones are proteins which control and regulate the folding of protein. So increased production of chaperones ensures that newly synthesized proteins are properly folded. Moreover, this signaling also reduces the protein synthesis, and when protein synthesis is decreased, it means that it tries to ensure that the proteins, the whatever protein is produced is properly folded, and the excessive increased protein rate is decreased to ensure that protein folding is proper. Moreover, which proteins are not properly folded, their degradation is increased. So this is ultimately called adaptive unfolded protein response. What is adaptive unfolded protein response? It means unfolded protein inside the endoplasmic reticulum bind to the signaling receptor, and there is signaling. When there is signaling, chaperone production increases and protein synthesis decreases, and ultimately they reduce the amount of misfolded protein, and misfolded protein degradation increases, and ultimately there is reduction in the load of misfolded protein inside the endoplasmic reticulum. This is adaptive unfolded protein response. However, if there is severe ER status or severe accumulation of misfolded protein, what happens? A signaling pathway activates the BH3 receptor. What are BH3 receptor? These were basically the apoptotic receptor. They induce back and backs, and they induce the activation of cospaces, which lead to cellular death. This is called terminal unfolded protein response. So this was the endoplasmic reticulum stress, and all these things are explained here in the text. Next, to move on. There's multiple diseases which can be due to this ER stress and misfolded proteins. These diseases, some of these diseases are due to misfolded protein which is totally lost, and there is totally loss of function, or they may be due to misfolded protein which are destroyed due to their deficiency, or misfolded protein that result in ER stress and cellular death, or they may be combination of both. So here are some example of disease. For example, cystic fibrosis is due to CFTR protein, familial hypercholesterolemia is due to LDL receptor protein, Tay Sachs disease due to hexokinase B subunit. So these three are diseases in which mutant protein is degraded, and overall there is protein deficiency. Next here are three diseases: retinitis pigmentosa, Creutzfeldt-Jakob disease, and Alzheimer's disease. These are due to misfolded protein that induce the cellular damage or cell loss. So these diseases were due to loss of functional capability of protein and decrease in protein, and these are due to loss of function due to ER stress induced cellular death. And these are the diseases which are combination of both cell loss and functional deficiency of protein, which is alpha-1 antitrypsin deficiency. So these are example of some diseases you can read here. Next, move toward the next factor, which is DNA damage. And DNA damage can be due to chemotherapeutic agent, can be due to radiation, can be due to viruses, and all these DNA agents, all these damages can lead to mutations, and mutation can transform the cell into the neoplastic cell or can lead to cellular death. Here example of P3 protein. P3 protein is basically a guardian of genome. What happens here? It basically involved in regulating the cellular cycle. If there is some damage to DNA, it arrests the cell cycle and induces the DNA repairing mechanism. If damage is mild and it is repaired, then cell cycle continues. But if the damage is not repairable, if it is too severe, then it induces the apoptosis of the cell. So these are the topics which we will study in the neoplasia chapter next one. So next one come toward inflammation. Inflammatory cell, as we know that when there is inflammation, there is a recruitment of leukocytes, macrophages, neutrophils. All these in addition to removing the damaging stimuli, they can also induce damage to the normal cells. Next come toward what are the common events in cell injury. So there are two basically common events. One is mitochondrial dysfunction, another is dysfunction in membranes. When there is mitochondrial dysfunction, there is a reduced production of ATP, and there is also when there is reduced production of ATP, it have multiple effects. There is also reduced capability to reduce oxygen properly. As a result, there is production of reactive oxygen species, and damage to mitochondria also produces high conductance channel in the mitochondria, and these conductance channels lead to loss of their proper membrane potential and compromise their function more and more. Moreover, mitochondria also have cytochrome C, which leak out into the cytoplasm and activates cospaces and lead to apoptosis. So mitochondrial dysfunction leads to loss of ATP. It leads to loss of proper reduction of oxygen and increases production of reactive oxygen species. It leads to the loss of proper membrane potential of mitochondria, which further compromises the function, and it leads to the release of cytochrome C into the cytosol, which leads to the apoptosis. Further defect in membrane permeability. So what type of membrane are altered? Mitochondrial membrane is damaged, plasma membrane is damaged, and lysosomal membrane is damaged. When mitochondrial membrane is damaged, ATP production declines, and it have multiple effects which we have discussed previously. When plasma membrane is damaged, there is imbalance of fluid and electrolyte. As a result, water enters the cell swells up, and so on. When there is injury to lysosomal enzyme leak out, and when lysosomal enzyme leak out, they damage the cell and surrounding cells and induce inflammation, which further damages the cell. So these were the common mechanism, and here we have covered the topic. So what we have discussed till now, here we in this lecture we discuss some common facts related to the cellular injury. What happens? Cell injury depends upon type of cell, type of injury, as well as injury result leads to some chemical and biochemical abnormality in the cell. Then we discuss different factors which are responsible for the cellular injury, which include hypoxia and ischemia, ischemic reperfusion injury, reactive oxygen species, oxidative stress, toxins, endoplasmic reticulum stress, and DNA damage, inflammation, and common events. All this was about today's lecture. Thank you.